Hello everyone! Today we are going to work on learning about cellular respiration. So I know you've already went over this in a good bit of detail in your lesson module so far, but I find some students really find it beneficial when people take the time to just draw out each step-by-step process within this grander process of cellular respiration. So just a warning, this is going to be definitely one of the longer videos that you're going to find in your module. However, a lot of students have said that going through this step-by-step is helpful. So let's get into it, shall we? A couple of things that I want to clarify before I start drawing stuff out. I'm going to be marking all of my enzymes in the color green. So green are our enzymes. Remember, enzymes are things that catalyze chemical reactions. And I'm going to be using our different substrates in the blue color. So we're going to be starting with glucose is where we're going to start, but as we go through the process, you'll see that glucose quickly gets converted into a litany of other things through different chemical processes to help us end up producing energy. And then we have arrows throughout, right, to indicate movement in our chemical reaction series. Now, something that a lot of people forget to go over are the differences between these two arrows, and they actually mean different things. If you see an arrow that is unidirectional, that means that the reaction that's taking place cannot go in reverse. It can only go in one direction. However, if you see a double-sided arrow, one that can go back and forth, these are what we call equilibrium reactions. And so these can go back or forwards, and it's all dependent on the concentration of the substrate, all right? And so I'll give you an example as we go through this. Let's start from the top with cellular respiration. You, as an example, you ate food, and in your food is glucose, and your body is now going to use that glucose that you ate to produce energy. So as like a brief recap, where's one of the sources that you can get glucose? Down the line, either directly from plant matter because you ate plants, or you ate something that ate plants, right? So photosynthesis is a large contributor to us providing us with glucose, right? So we're starting out with our glucose molecule here, and we're starting out in the bloodstream. So this is after, digestion and stuff has taken place, and your food particles have been broken down into their small subunits until we are left with little glucose molecules, all right? So in your bloodstream, we have glucose molecules, and the glucose is going to enter into a cell, leaving your bloodstream and entering into a cell. So this is my plasma membrane of a cell. So here we're in the extracellular space, right? Because this is outside of the cell. Inside of the cell in here, this would be our cytosol. So let's go ahead and label when the glucose enters, it enters into the cytosol of the cell initially. And it enters via a receptor. And this receptor in humans is triggered by insulin, right? So when insulin binds to its receptor, it triggers a response that ultimately allows glucose to enter the cell. Boop. So now we have glucose in the cytosol of our cell. So insulin triggers glucose transporters. Sound good? So here's our glucose, little diamond guy in the cell, in the cytosol of the cell. And now that glucose is in the cell, the first step that happens in cellular respiration, once we're inside the cell, is glycolysis. So remember, glycolysis comes in two steps. We have a step that requires energy to convert our glucose molecule into something else. And then a second phase is the energy producing phase, where we then gain energy moving through our series of steps. Sound good? So step number one, we have our energy requiring phase. So that means that ATP is being used up for this phase. So the first thing that happens is a reaction that is carried out by a kinase. It's hexokinase if it's outside of the liver, or glucokinase if we're looking at a liver cell. You do not have to know that level of specificity, just know that it is a type of kinase, okay? The specific names I'm not gonna harp you on, as long as you know the kind of enzyme is a kinase. And this is important for you to know because all kinases are a byproduct of the conversion of ATP to ADP. Or the reverse of this. Really what a kinase does is it moves phosphate groups around, and this usually either produces or uses up energy, okay? So here, basically what happens is the kinase will take one of the phosphates. Remember, adenosine triphosphate, so we have an A and then 3 P's attached, and that's an ATP molecule, right? So basically what a kinase does is it takes one of those phosphates and tacks it on to whatever chemical it's reacting with, all right? So now we have something called glucose 6-phosphate, and that cost us energy, making that glucose 6-phosphate. And the reason that it's called glucose 6-phosphate is not because it has 6 phosphate groups, it's called that because that is the carbon that the phosphate is attached to. It's on the 6th carbon of the glucose molecule. Sound good? And for any of you who like to go above and beyond, and you have heard about the term glycogen. Remember, glycogen is something we talked about way back when in Unit 1. Glycogen is how animals store their energy, right? That's how they can store glucose to be used later, right? This is where we get glycogen from, actually, is a side reaction can happen here if there's a lot of glucose 6-phosphate building up in the cell. Through a different process, through different enzymes, glucose 6-phosphate can be converted into and stored as glycogen. Sound good? Okay, so that's that. That's just step one now of the energy requiring phase. So let's keep moving forward. Once we have glucose 6-phosphate, we have an equilibrium reaction. So we're going to break down what that means in a second. And that equilibrium reaction is carried out by an enzyme called isomerase. again, we can tell that this is an enzyme because it ends in the word ase. And I will not require that you memorize that name, all right? The ones that I really want you to memorize are the ones that, what kind of enzyme we're dealing with, all right? If there's a specific type, such as a kinase. Sound good? So, isomerase will take glucose 6-phosphate and basically convert it into fructose 6-phosphate. Basically, it rearranges glucose 6-phosphate. Sound good? So, the key thing to note here is this nice double-sided arrow, all right? That double-sided arrow tells us that we are dealing with an equilibrium reaction, which means that the concentration of glucose 6-phosphate, if it's higher than the concentration of fructose 6-phosphate in the cell, then a cell will use isomerase to convert glucose 6-phosphate into fructose 6-phosphate, all right? But if the cell were to have a higher concentration of fructose 6-phosphate, then isomerase would actually do the reverse of this, and it would convert fructose 6-phosphate back into glucose 6-phosphate, all right? So the increase of glucose 6-phosphate is what drives this reaction forward, okay? So we need that increase in glucose 6-phosphate to drive the reaction forward, moving further down into cellular respiration to produce energy. So next, now that we have our fructose 6-phosphate, we have another enzyme coming in, and this is called phosphofructokinase. Okay, phospho relating to the phosphate group, fructo referring to the fructose aspect, right? And then kinase. Kinase is the big thing, right? So we're dealing with something that is going to require ATP. So we're going to be using up ATP again. So that way it can steal one of those phosphates and tack it on. So now our ATP is ADP. It's going to take one of those phosphates from ATP and tack it onto our fructose 6-phosphate to make fructose 1,6-phosphate. All right? Again, the 1 and the 6 are just referring to where in the molecule we will find those phosphate groups. And I apologize, I misspoke. It's actually fructose 1,6-bisphosphate, right? Because there's 2 phosphates. So the Bi is because there's 2 phosphate groups. Sound good? So fructose 1,6-bisphosphate. Here's where things get kind of crazy. So at this point, before moving forward too much, let's zoom out a bit and assess what we've done so far, all right? You ate food. The food was broken down into glucose molecules. Well, the carbohydrates were at least, right? Broken down into glucose molecules. Insulin helped allow that glucose to enter the cytosol of your cell, where glycolysis was able to take place. Just the first part of glycolysis. We haven't done the second part yet. And so at this point, we have used up 2 ATPs, right? 1 ATP was used up here, 1 ATP was used up there. So now we're moving on to the second phase of glycolysis, which is our energy producing phase, right? Energy requiring phase refers to using up energy. Energy producing phase is exactly what it sounds like. We're now going to start making energy, okay? Now this is the part of the glycolysis that gets a little bit complicated, so bear with me, all right? So going down to our fructose 1,6-bisphosphate. What happens here is something that we call the triose, because it's basically a 3-way reversible chemical reaction. So we have fructose 1,6-bisphosphate can be converted into, reversibly converted into DHAP, as well as glyceraldehyde 3-phosphate. Some people call this G3P. The important thing to note about both of these molecules, DHAP and G3P, is both of these are 3-carbon molecules. Going all the way back up here, I know I drew our little glucose guy as a diamond, but really remember glucose is a 6-carbon molecule, right? So it's 1, 2, 3, 4, 5, 6. So once we get down here, we have effectively cut this molecule in half, thanks to an enzyme called aldolase. So we have 2 3-carbon molecules, and each one of these goes through a series of steps. Basically what happens, let's start with glyceraldehyde, because I think that'll make things a little bit easier. So starting with glyceraldehyde, we have a reversible reaction. So it's driven by the concentrations of each molecule on either side of the reaction in the cell. We have a type of enzyme called glyceraldehyde 3P dehydrogenase. The important part of that name, it is a dehydrogenase enzyme, all right? G3P dehydrogenase. So knowing about dehydrogenases are important because these enzymes catalyze the oxidation of a substrate through the transfer of hydrogen to an acceptor. So let's see what this looks like for us. In this case, we are dealing with NAD+, and then dehydrogenase, G3P dehydrogenase, will basically snatch a hydrogen and donate it to this acceptor molecule NAD+, forming NADH, all right, which is a source of energy, and producing from this, it converts glyceraldehyde 3P into 1,3-bisphosphoglycerate. From here, phosphoglycerate kinase, right? So we're dealing with another kinase and another reversible reaction. Phosphoglycerate kinase does what kinases do best, but this time, instead of stealing phosphate group from ATP, it steals the phosphate group from 1,3-bis-phosphoglycerate. So now the kinase is acting to convert ADP into ATP. Does that make sense? So it stole the phosphate instead of from ATP, it stole it from 1,3-bis-phosphoglycerate, converting ADP into ATP, right? So that's another source of energy. I'll go ahead and put little things so we can tell which guys make energy. Sound good? This process then converts 1,3-bis-phosphoglycerate to now being 3-phosphoglycerate. If we want to break down where this name came from, basically the kinase here stole one of the phosphate groups on that first carbon there and then gave it to ADP, making that ATP, right? So now it's not a biphosphoglycerate because bi refers to 2, it's just phosphoglycerate. And that phosphate group is on the third carbon of that molecule. Sound good? Okay. From here, 3-phosphoglycerate is then converted into PEP via an enzyme called mutase. And then we have our final step in glycolysis for this half of the triose, where PEP is converted into pyruvate, which is, again, a 3-carbon molecule. And this is important because this is a unidirectional reaction. So once we get to PEP, if pyruvate kinase, all right, so another kinase, if pyruvate kinase interacts with PEP, it will convert it into pyruvate, and that will only go in one direction. It can't be converted back into PEP. So just like before, this kinase will also be taking a phosphate group from the substrate here to convert ADP into ATP. More energy. Big yays. All right? So that is this half of the pathway past the triose in glycolysis. Now, what happens over here? Let's talk about it. Essentially what happens here is DHAP can actually be converted into glyceraldehyde 3P or G3P. So basically everything, pardon, that's my blind cat. He'll be okay though, I have him right next to me. Anyways, DHAP can be converted into glyceraldehyde 3P. And this is actually also a reversible reaction. So let me go ahead and fix that really fast. So the arrow is double-sided here. So basically everything that we saw happen over here, it happens over here as well. So basically for every 1 molecule of glucose that goes into this system, we end up with 2 pyruvate molecules, which are 2 3-carbon molecules. And we also end up with 2 ATP molecules. And we also added into the system an NADH molecule. And that's just for this side, right? The same thing happens over here as well, right? Whoop. So that actually means we have a total of 4 ATP and 2 NADHs by the end of this part of the process. Does that make sense? So that is glycolysis, and again, this all takes place in the cytosol of a cell. Now that we have our pyruvate molecule, there are a couple of options, all right? Option number 1 is if there is no oxygen around, so that would mean we are in anaerobic conditions, then pyruvate would actually get converted into something called lactate. People also commonly refer to lactate as lactic acid. Your muscles ever burn after you do a heavy workout, right? That's lactic acid burning in those muscles, right? Because your cells have been depleted of oxygen. So when they need to make that energy, they tend to just stick with this pathway where they convert pyruvate into lactate. Now, converting pyruvate into lactate requires energy. So I'm going to actually move this statement so that way we have room. Basically what has to happen is NADH is used as an energy source to fuel this reaction, leaving us with NAD plus and lactate, or also called lactic acid. Sound good? And then in the liver, you don't need to know this part, but just for your own edification, if you're curious, in the liver, lactate is actually used to then make more glucose. So that way, later, when you do have oxygen, you can use that glucose to generate a lot of energy when there's oxygen around, all right? But this is the no oxygen environment model of how you can at least get a little bit of energy, right? You get a net total of 2 ATPs and 2 NADHs, which honestly, it's better than nothing, right? So that's good. But it's not nearly as good as having oxygen around where you can actually carry out the rest of cellular respiration in the citric acid cycle. So some of you may be wondering, where does this NADH over here come from? And that actually comes from this guy up here. So NADH that was produced up there actually gets used, and that NAD plus can be used then again to restart this cycle and act as an acceptor of that hydrogen later in the process. Does that kind of make sense? So it's all a crazy awesome cycle. Sound good? Now, what if there is oxygen? If there is oxygen, this 3-carbon molecule pyruvate, these guys will go on to enter the mitochondria. Remember the mitochondria? It's an organelle inside of the cell, so we're still inside of the same cell. And the mitochondria has a double membrane, right? So membrane number 1, membrane number 2. So here, right here is inside of the mitochondria. And so pyruvate, if oxygen is present, so if yes, oxygen, then pyruvate will go into the cell.
And in one of your videos, we actually talk about how it goes into the mitochondria through a series of channels.
And it is in the mitochondria that the citric acid cycle, I should say the citric acid cycle, which is why a lot of people call it the TCA cycle. And then other people will also call this the Krebs cycle, named after the guy who came up with this or who discovered this. Ain't it always? All right. So either way, you call it is fine, as long as you know you're talking about the same thing. This is what happens in the mitochondria if oxygen is present, all right? And the cycle occurs one pyruvate at a time. So we're actually going to just focus on as one pyruvate at a time, and then we'll do math later. Sound good? All right, so we have pyruvate inside of the mitochondria. Yay! So the citric acid cycle is ready to go. Pyruvate, once it enters, it can go in 2 different directions, all right? Direction number 1 that it can go, and notice that this is a one-direction arrow. This means that it's a non-reversible reaction. So one way that it can go is it can be converted into acetyl coenzyme A, and this is a 2-carbon molecule. So how does this happen? This happens by NAD plus working to pick up and convert into NADH, and CO2 is also lost in this reaction. And so that's why this molecule of pyruvate, which was a 3-carbon molecule, has gone down to a 2-carbon molecule, because one of those carbons was let go through the release of carbon dioxide. And this process is done by, you might have figured it out because you see NADH, right? This is done by a dehydrogenase enzyme. Specifically, and this name makes sense, pyruvate dehydrogenase. Do I have room? Well, that's good enough. All right. Sound good? So this is where we have our first loss of carbon, 1 lost carbon. The other direction that pyruvate can go when it enters into the mitochondria is it can also go through another non-reversible reaction via pyruvate carboxylase, which converts pyruvate into something called oxaloacetate. This is really important for the cycle. This right here is a 4-carbon molecule. Hence the name of our enzyme. It's called a carboxylase because it adds a carbon to our molecule, all right? So in one direction we're losing a carbon, in the other direction we're gaining carbon. Both are absolutely critical to the citric acid cycle. So let's keep it going. The way that the cycle goes is actually in this direction. But this step right here over to the left is very important because it actually contributes to step 2 or 3, depending on how you look at it, in the citric acid cycle. So let's break it down. We have our acetyl-CoA, which is a 2-carbon molecule. And the next step in the cycle is to actually convert acetyl-CoA into citrate. And citrate, hence the name citric acid cycle, citrate is a 6-carbon molecule. And to get acetyl-CoA to somehow magically become citrate, you need 4 more carbons, right? Where can we get those 4 carbons? Oh, I know, we can get it from over here, right? So that's how it comes into play. And just to make things super clear, pyruvate can't be both oxaloacetate and acetyl-CoA. The point of this diagram is to show you that pyruvates are constantly entering the mitochondria because you are eating glucose, and glucose is constantly going through the cycle and constantly entering mitochondria, right? So some pyruvate will go to acetyl-CoA, some will go to oxaloacetate, and them coming together is what helps produce citrate, our 6-carbon molecule, which is where our cycle gets its name, the citric acid cycle. Citrate, a lot of people also call citric acid, just as an FYI. So we have our 6-carbon molecule, and now we're entering the reversible phases. So the phases that are at equilibrium and are controlled by the concentration of each substrate, right? Substrate or product, depending on how you look at it. So citrate can then be converted into isocitrate, and this is via an enzyme called aconitase. Do I care that you know that? Not really. Just remember ACE means enzyme. It's doing things. Cool? All right, so this is still 6 carbons. It basically just rearranged the carbons. All right, rearranged the molecule a bit, but still 6. Next, we have a unidirectional reaction where isocitrate is converted into alpha-ketoglutarate. Alpha-ketoglutarate, though, is a 5-carbon molecule. And so what happens to get isocitrate to alpha-ketoglutarate is we have energy produced in a process. So NAD plus picks up a hydrogen, or a hydride, right, and which produces NADH. And we lose another carbon through CO2. So there's where we lost the carbon that was there. It was lost to CO2. So can you guess what enzyme probably did this, or what type of enzyme? If you had to choose between a kinase, a carboxylase, or a dehydrogenase, what likely is responsible? Right, this is a dehydrogenase enzyme, okay? Specifically, it's called isocitrate dehydrogenase, which makes sense because it specifically dehydrogenizes, if you want to call it that, isocitrate into alpha-ketoglutarate. So here, I'm just going to go ahead and put some jazz on the NADH to help us keep track of the fact that we have now produced energy, or a source of energy. And then over here, I'm also going to mark that we lost one more lost carbon, hence why we have 5 carbon molecule right here in alpha-ketoglutarate. All right, so we are now halfway through the cycle. You got this, guys. I believe in you. And hopefully this is kind of making sense that you can see some semblance of a pattern in how this works, all right? So the next step is alpha-ketoglutarate is then converted into succinyl-CoA. And so how does that happen? So succinyl-CoA is a 4-carbon molecule. A similar process as to what you just saw before happened. So let's see if you can fill in the gaps on your own now, right? What was likely put in to this reaction to produce succinyl-CoA from alpha-ketoglutarate? Right, we used NAD plus as a acceptor of that hydrogen, so that forms NADH, right? And we lost another carbon, so that means that CO2 is likely released, which it was, so that's where we lost that carbon from. Boop. So what kind of enzyme is probably responsible for this reaction? Right, another dehydrogenase, okay? Another dehydrogenase is responsible for this reaction, and the name of the dehydrogenase is alpha-ketoglutarate dehydrogenase. See how the names kind of make sense? All right, so I'll go ahead and mark this guy as another lost carbon, so one more lost carbon. So we're halfway through our cycle, time to finish out the clock. So from succinyl-CoA, we produce something called succinate, and that is via an enzyme that is called thiokinase. However, I just want you guys to know that it's a kinase. So if we're talking about a kinase, what are you thinking of off the top of your head immediately? Right, kinases deal with phosphate groups. So normally this is where I tell you we are dealing with ATP and ADP. However, this is special. This is dealing with GDP and GTP. However, don't let this confuse you. They're basically equivalents of one another, all right? One GTP molecule can easily be converted into an ATP molecule, so you can think about it that way if you want to. Just know that technically we're dealing with GDP and not ADP here. So in essence, the moral of the story here is, boom, energy is produced in this step thanks to that kinase, which means that a phosphate group was taken and added to that GDP to produce GTP. From succinate, we have another reversible reaction thanks to a dehydrogenase enzyme, specifically succinate dehydrogenase, where another type of energy-producing molecule, or energy-contributing molecule called FAD, is converted into FADH, and this also is a source of energy, so I'll go ahead and make him, yay! All right, so getting that nice hydrogen ion, kind of like we do with NADH, and this produces a molecule called fumarate. Fumarate then undergoes a chemical reaction with an enzyme called fumarase, which is basically literally translated into an enzyme that deals with fumarate. So very creative naming, guys, right? So fumarate via an enzyme called fumarase is converted into malate. And if the concentration of malate is high enough, then that reaction can reverse and fumarase can convert malate into fumarate again. But anywho, fumarate converted into malate to continue the cycle. And then, this is where it gets interesting, we have the last stage in our cycle where malate can be reversibly converted into oxaloacetate via malate dehydrogenase. And so we're dealing with a dehydrogenase again. So just like before, that means we are dealing with NAD plus being converted into NADH. Boom, 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 boom. All right? So here is our whole cycle, all right? And we were just focusing on what happens if we hone in on one pyruvate molecule. So from one pyruvate molecule, this whole cycle occurs. So let's go ahead and count up what we have, all right? From one pyruvate molecule, we have some NADHs, some GTPs, and some FADHs, okay? So let's count them up, shall we? For NADHs, we have 1, 2, 3, and 4. And again, that's just from one pyruvate molecule. So we have 4 NADHs. And then for GTPs, whoop, we have one of those. And FADHs, we have one of those. All right, so we're going to hold on to that for later. I'm going to go ahead and write it down from one pyruvate. Whoop. So now this is where things get interesting, all right? All of these energy sources then go to something called the electron transport chain. We're moving on from the citric acid cycle to something called oxidative phosphorylation, which deals with the electron transport chain and chemiosmosis, all right? So where does that take place? Do you guys remember where the electron transport chain occurs? Right, it takes place in the mitochondrial membrane, right? All of these products here are going to travel, all of these energy products are going to travel here, where we have the electron transport chain, all right? So I'm gonna go ahead and draw my little dotted lines. These guys are all gonna go, boop. So off to the electron transport chain we go. Now I am going to go ahead and blow up this image down below this drawing here. Sound good? Okay, zooming in on the electron transport chain in our intermembrane space. So just as a point of reference, I'm gonna go ahead and draw out this square that we're zooming in on. And then what we have is, right here is our mitochondrial membrane that we're dealing with, where all of our complexes that are a part of the electron transport chain are found. But just for a point of reference, the whole structure of all of this can be confusing to some students. I just want to take a second and zoom out a bit. Just as like a brief reminder, here's a mitochondria. And now the mitochondria has an outer membrane like this. So this space in here, all that's the outer mitochondrial membrane. And then inside of the outer mitochondrial membrane, we have something that looks like this. And so this whole thing, that's the inner mitochondrial membrane. And so the space that's in between the outer membrane and the inner membrane, this whole area here that goes all the way around, all of this blue, that is the intermembrane space. Remember, inter means between. Sorry, I ran out of room there. But inter means between. So this is the space between the outer inner membrane. So this is the intermembrane space, okay? So over here in our square, all right, this is a mitochondrial membrane. Specifically, this is the inner mitochondrial membrane. And out here, this is the intermembrane space. So down here, this is inside of the mitochondria. So this is what we call the matrix of the mitochondria, or the mitochondrial matrix. And so referring back to our image over here, all of this jazz in here, that is the matrix. It's the inner part of the mitochondria. Sound good? All right, so this is the mitochondrion. Outside of the mitochondrion, we have the cytosol and all the other organelles that are floating around. Make sense? Okay, so now that we have a little bit more orientation, let's go ahead and dive into the electron transport chain, all right? So zooming in, we now have, thanks to the TCA cycle, a bunch of goodies to work with to help us produce some energy, right? So from the citric acid cycle, we were given several electron carriers, such as NADH and FADH. Ooh, can I spell? So those came from the TCA cycle. And now it's time to see how these guys that are each carrying electrons with them, how these guys are then going to donate their electrons to the electron transport chain in the effort to produce energy. So along the electron transport chain, we have several complexes. We have complex number 1, complex number 2, complex number 3. We have a couple of helper guys as well. And then we have complex number 4. And then at the very end here, we have the big bopper. We have ATP synthase. And if it has synth in it, that probably means it's synthesizing something. So yes, this is where ATP is synthesized or made, all right? So the goal is to get all of our electrons down the line so we can use them to help produce ATP, or energy for the cell. So let's break down how this happens, starting with NADH. NADH carries 2 electrons, all right? And it donates those electrons to complex number 1. And this process produces NAD+ as well as a proton. As the electrons move through this protein complex, energy is gradually released, and this energy is used to pump hydrogen ions, or protons, that are inside of the mitochondrial matrix into the intermembrane space. The electrons are then transported by this special little molecule here towards their next destination, to complex number 2. And it's here that electrons are actually donated by FADH2, which produces FAD as well as 2 protons, because it's FADH2. And then all of these electrons are then passed on to another protein complex. At this point, this next protein complex, the electron energy is used to once again transport hydrogen ions across the membrane and into the inner membrane space. So you might have noticed then that suddenly we have a pretty darn high concentration of protons or hydrogen ions in this intermembrane space. This will be very important soon, so keep this in the back of your mind that we are increasing the concentration of protons in the intermembrane space, all right? That concentration is becoming pretty darn high as electrons are being passed down this process. Now the electrons are transported to a final protein complex, where their energy is used to once again pump hydrogen ions into the intermembrane space, making the concentration even higher. Now the electrons need somewhere to go, a final destination to complete their journey. And so here's where oxygen comes in, right? You often hear of oxygen as the final electron acceptor, and this is why. So this is why oxygen is so important and why it's a necessary part of this specific process. Oxygen, not technically O2. So oxygen at this point will come in and accept electrons as well as some hydrogens. So oxygen comes in and accepts the electrons as well as hydrogens from the matrix, making water. This is one of the main reasons why your cells need oxygen, to accept these electrons, or the electron transport chain will grind to a halt. Now it's time to touch on the last part of the process where we actually get to synthesize some ATP. So remember that the concentration of hydrogen ions we've been building up in the intermembrane space? Well, now it's time to use it to make ATP, a protein called ATP synthase that we have over here. That's my cat again. So here we have ATP synthase. This makes a channel that allows hydrogen ions to pass through from high concentration of hydrogen ions to a lower concentration of hydrogen ions. Protons, like everything, naturally want to travel down their concentration gradient from an area of high concentration to an area of low concentration. Remember going through that earlier in the semester? Yes, so same rules apply. So this is a process, remember, known as diffusion. But since they have to travel through ATP synthase to get back into the mitochondrial matrix, we would call this facilitated diffusion, the passing of those protons through a channel into the mitochondrial matrix. So the facilitated diffusion of these hydrogen ions through ATP synthase actually causes the ATP synthase to spin like a turbine and produce, from this process, ATP. So since we made these since we got these many electron carriers from the citric acid cycle, right, cellular respiration, the electron transport chain happens over and over again. So from this process, we ultimately produce anywhere between 30 to 36 ATP molecules in total. And so if it helps you to think more about this mathematically speaking, you can think about it this way in terms of the electron carriers and their equivalence to how much ATP they produce in the electron transport chain. So roughly for every one NADH molecule, you get about 3 ATP. For every FADH molecule, you get about 2 ATP. And then for every GTP molecule, you get about 1 ATP. And then obviously 1 ATP is equivalent to 1 ATP. So if we want to scroll up and count everything up to make sure it all makes sense, over here from 1 pyruvate, we got 4 NADH, 1 GTP, and 1 FADH. So that would mean that for 2 pyruvate, so for 1 glucose molecule, you end up with 2 pyruvate. So if we wanted to look at this from the perspective of a single glucose molecule, we would actually have 8 NADH, 2 GTP, and 2 FADH. And so that would mean that we would essentially have 8 times 3 ATP from the electron transport chain, which would be 24 molecules of ATP. For GTP, 2 times 1 is 2. Hello, Smudge. That's the blind cat. He's okay. He's sitting right here with me. And then we have for FADH, 2 times 2 is 4. And so adding this all together, we end up with 30 ATP. But remember, this is just from the citric acid cycle. We also need to add in the energy that we are going to get from glycolysis, right? So zooming out a bit further, from glycolysis, we got 2 ATP and 2 NADH. So from this, we get 2 ATP, as well as 2 times 3 for NADH to ATP, and that would be 6. 2 plus 6 is 8.
Now, I know what you're thinking. 30, last we checked, plus 8 is equal to 38, not 30 to 36, right? Well, this is because what we are looking at here is in a perfect system where no energy is actually used to help facilitate the movement of electron carriers, right? So 2 things to keep in mind are, number 1, the system we are looking at is within a living system, right? This is within living cells. And things that happen in life in general don't happen flawlessly, right? Nothing is 100% energy efficient. The other thing to keep in mind is that to make energy, we tend to need to spend energy. A perfect example of this is something that we see in glycolysis, right? We see that we have an energy requiring phase where we used up 2 ATP just to get this whole process started. And the same holds true if we want to transfer molecules like NADH and FADH, right? If we want to transport these electron carriers to the electron transport chain for oxidative phosphorylation, that actually costs energy. And on average, it usually costs around 2 ATP to transfer these molecules, but it can take more because remember, like I said, this is not a flawless system to bring these molecules to where they need to be. So this is why whenever we study the production of ATP through cellular respiration, we provide a range of ATP we would expect to get after this whole process has concluded. Make sense? So on that note, I want to actually now synthesize this in a way that hopefully helps people keep all these numbers and procedures in mind, okay? So over here, I'm gonna go ahead and erase 30 plus 8 is 38, because I think you know that. So keeping in mind that if this were a perfect system and we got maximum ATP yield, we would get 38 ATP from the system. However, that's not very realistic. Now over here, I'm going to draw out a table for us, and we're gonna fill in this table together to summarize everything that we've gone over in this procedure of glycolysis and the Krebs cycle and oxidative phosphorylation, including the electron transport chain, together so far. So we are going to go over 5 key topics in this table that we're going to fill in together. The first is going to be the source, which basically refers to which stage in the process we are referring to within cellular respiration. The next is going to be carbon flow. So remembering that energy and matter can neither be created nor destroyed, right? It must always be accounted for in a closed system. So since this is a system where we are dealing with matter, we must always be able to account for the different components of this cycle. So specifically, we are keeping track of carbon. So we're gonna look at carbon flow to make sure that the numbers balance out. If 6 carbons go into the system, that means 6 carbons have to be there at the end of this whole process, right? All matter must be accounted for. Next, we're going to go into substrate level phosphorylation. So keeping in mind that if we're talking about substrate level phosphorylation, we're talking about specifically the phosphorylation of ADP to ATP. So this is us looking into direct sources of ATP in cellular respiration. So where do we get energy without needing to use the electron transport chain, right? Where do we get ATP from the system without having to use oxidative phosphorylation? And then after that, we will look into oxidative phosphorylation, right? Here, this is where we will look at where we get, or how many ATPs we get from our electron carriers that have brought electrons to the electron transport chain for oxidative phosphorylation. So again, the phosphorylation of ADP to ATP, but this is the phase that requires oxygen as that electron acceptor. And so here, this is going to tell us how much ATP we get from that last stage of cellular respiration. And then this final column is where we will write out the ATP maximum yield. So this is where we'll do, our math will be in this column, and then we'll also have a little bit of math on the bottom where I'm going to actually put in the totals for everything. All right, so starting with glycolysis. In glycolysis, we started with glucose, which is a 6-carbon molecule. So we are starting with 6 carbons. And from glucose, through the litany of procedures that occur during glycolysis, we end up with 2 pyruvate molecules. And each pyruvate molecule is made up of 3 carbons. So far so good, right? 2 times 3 is 6. So all of our carbons in the system are still accounted for. We started with 6 carbons, and so far we still have 6 carbons total through 2 pyruvate molecules, each with 3 carbons. Next, we have what we sometimes call our transition reaction. And this is when we are converting pyruvate into acetyl-CoA right before the big part of that Krebs cycle starts, all right? So some people lump all this together, but a lot of times we like to really separate glycolysis and the citric acid cycle by saying that the conversion of pyruvate into those acetyl-CoA molecules is a transitional reaction occurring and bridging glycolysis to the citric acid cycle. So from our 2 pyruvate molecules, each that have 3 carbons, we get 2 acetyl-CoA molecules, right? Each pyruvate gets converted into an acetyl-CoA molecule, and each acetyl-CoA molecule has 2 carbons. And so now you're like, oh no, we don't have equivalent carbons, right? We went from 6 carbons from our 2 pyruvate molecules down to 4. So how can we account for all of our carbons? But wait, during this phase, we also released carbon dioxide, right? For every pyruvate molecule that is converted into acetyl-CoA, we lose 1 carbon dioxide. So for 2 pyruvate molecules, that would mean we're losing 2 carbon dioxide molecules. And there's 1 carbon molecule in every CO2 molecule, so 2 carbon dioxides would mean we're losing 2 carbon molecules, right? So we have 2 acetyl-CoAs, and each have 2 carbons. 2 times 2 is 4, plus 2 more is 6, so we're still on track. And next we have our citric acid cycle, right? The citric acid cycle. And in the citric acid cycle, where we have our acetyl-CoA, acetyl-CoA undergoes chemical reactions that are followed by many, many more chemical reactions to complete the whole cycle. But long story short, throughout this entire cycle, we end up releasing 4 carbon dioxide molecules. And so from this, if we wanted to keep track of all of our carbon that is going into the system versus coming out, so now we're looking at the total, we started with 1 glucose molecule, which is made up of 6 carbon molecules. And through this process, we released 6 carbon dioxide molecules. And each carbon dioxide only has 1 carbon. And so the net total of carbons balances out, right? We put 6 carbons into the system and we left the system with all 6 carbons accounted for. They were just released as carbon dioxide. And through this process of basically breaking down glucose into smaller and smaller molecules, we produced ATP. And so let's go ahead and move on to the next column of our table, starting with glycolysis, looking at substrate level phosphorylation. Remembering that this is where we are looking at the direct production of ATP, where we do not need the electron transport chain, but just how much ATP are we producing in the system before oxidative phosphorylation. Sound good? So in glycolysis, we produce a net total of 2 ATP per 1 glucose molecule. Remembering that even though we produced 4 ATP in total, we had to spend 2 ATP to get those 4. So net total, we have 2 ATP from glycolysis. Then in the transition reaction, we produce NADH, right? But we don't actually produce any direct ATP. NADH is an electron carrier, and so it has to go through the electron transport chain or interact with the electron transport chain in order for ATP to be produced. So no ATP produced directly from the conversion of pyruvate to acetyl-CoA. However, in the TCA cycle, we do get GTP produced, and GTP does not need to go into the electron transport chain. It doesn't need to undergo oxidative phosphorylation in order for ATP to be produced. GTP can be actually directly converted into ATP. And so for each cycle of the TCA cycle, we get 1 GTP molecule. But remember, 1 glucose molecule produces 2 pyruvate molecules, which means now we are dealing with 2 rounds of the TCA cycle. So that would mean total for 1 glucose molecule, we would be getting 2 GTP, which is equivalent to 2 ATP. So this would total up to 4 ATP molecules before oxidative phosphorylation even happens. Now, oxidative phosphorylation, though, as you're going to see, this is where we get the maximum amount of energy. So starting with glycolysis. From glycolysis, we get 2 NADH molecules. And once those 2 NADH molecules undergo oxidative phosphorylation, we end up with 6 ATP. Remembering that for each NADH molecule is equivalent to about 3 ATP molecules once they've gone through oxidative phosphorylation. So we get 6 ATP from those 2 NADHs that we got from glycolysis. Now moving on to the transition reaction, we also produced 2 NADHs, remembering that we are accounting for 2 pyruvate molecules, right? So a single conversion of pyruvate to acetyl-CoA produces 1 NADH, but this happens twice for every 1 glucose molecule. So for 2 NADHs, we would get, again, 6 ATP from those 2 NADHs. Now here comes the busy part, the TCA cycle. This is where we get a lot of our electron carriers, right? This is where we get the bulk of our NADH as well as FADH. So we have, from the TCA cycle, 3 NADH molecules per cycle, but remembering 1 glucose molecule results in 2 cycles. So 3 times 2 is 6, right? So we get 6 NADH molecules. And remembering that NADH is equivalent to 3 ATP, so 3 times 6 would be 18 ATP molecules from 6 NADHs. And then for each cycle, we produce 1 FADH molecule. But again, we're dealing with 2 cycles, so we are now dealing with 2 FADH 2 molecules. And for every FADH, we get 2 ATP about. And so that would mean we get 4 ATP from our 2 FADH molecules. Make sense? So now adding all of this together, I'm going to go ahead and highlight our different ATP amounts. So on the substrate level, we got 2 from glycolysis and 6 from oxidative phosphorylation, totaling 8 molecules. If we have maximum yield, that is, right? We're talking about perfect system, right? So if this were a perfect system, we would get a total of 8 ATP molecules just from glycolysis if we include the ATP that comes about through oxidative phosphorylation at the very end of cellular respiration. And then for the transition reaction, we get 6 ATP total. From the TCA cycle, we get 2 plus 18 plus 4, which totals to 24. And so adding all of these together, if you want to go down the line first for our oxidative phosphorylation, sorry, I forgot to add all those up for you first. Oxidative phosphorylation altogether, we have 6 plus 18 plus 4. That would be 34 ATP just from oxidative phosphorylation. And then if we want the grand total maximum yield of ATP, we add all these together. They should add up, as well as adding these ones together. They should add up to the same number if we did everything right, and that would be 38 in a perfect system. Now remembering that in reality, we're going to end up somewhere with between 30 to 36 because it costs energy to transport things and make energy. Sound good? So if you've ever heard that cellular respiration produces exactly 34 ATP,
That's probably because people are mistakenly only referring to the energy we get at the very end through oxidative phosphorylation in a perfect system. They're basically excluding the ATP we produce directly in this whole process. They're only looking at a part of the whole.
But the total maximum yield in a perfect system would be 38 ATP, which in reality will equivalent to about 30 to 36 ATP. Well, hopefully this discussion was really helpful to you, and I hope that this helped provide clarity to certain aspects of cellular respiration. Thank you so much for going through all this with me. We started with glycolysis, which happens in the cytosol of the cell, went all the way through the Krebs cycle inside of that mitochondrial matrix, and then ended with oxidative phosphorylation, the electron transport chain, in our mitochondrial membrane. This helped clarify things for you, and I wish you the best of luck moving forward.